Okay, so let's begin a series of 2 lectures we're going to have on the structure of the nervous system, or neuroanatomy. And this will give us a foundation for the knowing the certain structures we're going to need as we examine sensory and motor systems, and then as we look to more less defined systems like language and learning and memory later on. Having a map in your head, no pun intended, pun kind of intended, of where these structures are will help you as we begin to discuss the circuits and pathways that are going to be involved in those various brain functions. Now, the brain is an extreme, and the spinal cord, the central nervous system, is an extremely complicated structure. And we have by no means fully mapped out all the details about it and all its complexity. So what we're going to have here is a general overview of the major structures in the brain and spinal cord, and also an examination of some of the basic layout of the peripheral nervous system as well, because of course we'll need to understand the structure of the peripheral nervous system to see how it communicates with the brain and spinal cord. Your book begins with a comparison of the brains of various mammals. So this chart shows you different mammalian brains blown up so you can kind of see their structures and compare them. And then over here shown with their relative sizes. And note that rat, that the centimeter scale there going from rat to dolphin. And even though you can see very distinct differences between these brains of different mammals, what I want to emphasize is that the layout of all these is more or less the same. What we see here is, with the more elaborate looking brains, is just that elaboration on a theme and a pattern of development that's consistent. Now, again, we're going to be looking and focusing on mammalian brains. If we look at saurid brains like reptile and bird brains, we'll see some distinct differences. The most notable difference is in the cortex, the outer part of the cerebrum. In mammals, you have a part of the cerebrum known as the neocortex. And in mammals, we think of as being more intelligent, with higher development of their brain. It is this neocortex which seems to have become more elaborate. Notably, you can see that as you go from a rat to a cat and a human and dolphin, the neocortex is the part on the outside that becomes more and more folded and elaborated. The area of that neocortex increases, particularly its surface area. The book uses the brain of a rat a lot for illustrations. And there's a few reasons for that. First of all, a lot of the studies on basic neuroanatomy have been done with rats and similar animals. Second one, it's the simplest, I guess, visually for you to understand, especially given how the book is going to explore the structure of the nervous system using the development pattern as a pattern to talk about the different parts of the nervous system. And finally, the axis, and this is not true just of the rat, but of other animals with similar body plans. The neural axis of the rat is straight. You can see its cortex, cerebellum there, and the brainstem and the spinal cord kind of all lining along one plane. Not perfectly so, but in the human, everything's bent, right? You can just see the brainstem coming out of the bottom part of the human brain there. So everything is bent 90 degrees along the brain stem in the human, or approximately 90, it's not quite 90 degrees. And so when we kind of translate between human and brain, humans being a bipedal, and these other animals being quadrupeds, we have to kind of keep in mind that bending of the axis. So to keep it simple, as they discuss, your book kind of relies on the rat brain, drawings of the rat brain to show that. Now, along the way, I'm going to be throwing in pictures of human brains to kind of show you the correlation between those two. So the idea is presented in simplified form using the rat brain, and then I'll show you kind of the elaborations in the human brain. And so hopefully by the end of this 2 lectures, we'll have a fairly good overview of the overall structure of the nervous system. Okay. Now the second chapter in this series is an atlas, and I may draw pictures from that atlas here, or I may draw pictures from that atlas as we discuss the structure of the brain in later chapters. Okay, so before we get into this, we need to have a foundation of terminology. And this foundation is useful for any anatomical study, but is especially useful for the study of the nervous system in our case. And the terms I'm going to give you are terms that relate to quadrupeds and can be used to relate to humans. In humans, we tend to use some specialized terms like superior and inferior that you don't necessarily use in quadrupeds, but you can use them in humans. You can use the quadruped terms. You just have to kind of translate, again, that neural axis turn and imagine the human sort of walking on all fours with the nose stuck straight out in a quadruped configuration, and that'll allow you to kind of mentally switch, again, no pun intended, between those two kind of frames of reference. Okay, so what are some of the terms we're going to see related to this? Well, we have anterior, which means towards the front, and that is often referred to also as rostral, which means towards the beak or towards the nose. So in a human, those are the same, and in a quadruped, those are the same. Now, keep in mind that in a quadruped, towards the nose is towards the front of the animal, but in a human, towards the front may be towards the belly. Okay, depending on what part of the body you're looking at. So usually, when we say anterior in a human, we mean the same thing as ventral in an animal, right? But in the brain, that may be down. Okay, so let me show you these terms as it relates to the rat brain, and then hopefully when we get into the human brain, you'll be able to see the difference. So anterior means towards the front, rostral means towards the nose, posterior means towards the back, caudal means towards the tail, dorsal means towards the back, ventral means towards the belly. The midline is the line of symmetry in a bilaterally symmetrical animal. We'll see that in a moment. Medial means closer to the midline. Lateral means further away from the midline. Ipsilateral means on the same side of the midline. So two things that are ipsilateral are both on the right side of the midline, for example. Contralateral means on opposite sides of the midline. Okay. Now these two are especially important when we think about how information flows through the nervous system in pathways that may stay on the same side of the midline or may cross the midline. And then we have a series of discussions of planes, right? The brain being a complicated three-dimensional structure, and the media that we have to communicate about it still being largely two-dimensional. Books, images on a computer screen, still largely two-dimensional. So the way that this is being has traditionally been conveyed is by literally or in the imagination slicing the brain up into planes and presenting views along those planes. And we will see different views along different ones of these planes. So the three main, or what we call orthogonal planes, are sagittal, horizontal, and coronal. And then the mid-sagittal, meaning the sagittal plane drawn along the midline, has a special significance that we'll see. Okay, so here I've just been throwing up terms. Let's see them in action. Okay, so here we can see dorsal versus ventral, caudal versus rostral, or anterior versus posterior. Okay. Then we have, from this view, we can see the midline, and we can see lateral and medial. Now this shows us some of the sections. This is a sagittal section. In particular, this is a mid-sagittal section. We can see the sagittal section in the rat is drawn along the rostral to caudal and dorsal to ventral axis, or a plane that's drawn along those axes. And the mid-sagittal section divides along that midline, the sagittal section dividing along that midline. Now, we can also take a sagittal section away from that midline, and we'd call that a parasagittal section. So it's parallel to the mid-sagittal section. Here's a horizontal section, and then this is a coronal section, or sometimes called a frontal section. Okay, now to show you how this might differ in a person, let me find a good picture. All right, so this is a picture that's from later in the chapter, but notice, of course, the human, the spinal cord goes up, and then the brain turns 90 degrees. Okay, so what would be dorsal in the brain is top, but in the spinal cord it's back. So we're just going along the back, going along the back, going along the back, but now the whole thing turns 90 degrees, and so now this is towards the back, or dorsal. What is towards the front here is the same thing as ventral, which is towards the bottom of the nervous system. So in a human, anterior is the same as ventral, and dorsal and posterior are the same for most of the body. But when we get into the brain, dorsal becomes superior up, and ventral becomes inferior or down. And now anterior and rostral are the same. Okay, so it's all about understanding how things change when that axis of the nervous system turns. But that being said, we are going to be mainly focusing on the brains of quadrupeds here, the rat, and so we can think of these, we can just simply use these quadruped terms, and we can simply imagine a human kind of pretending to be a four-legged animal, like a little kid pretending to be a dog or something, and holding its body in that position, and that'll give us the analogous terms. Okay. Now, what I've been referring to is a common position for understanding these terms called the anatomical position. So the confusion again comes because the anatomical position for a quadruped is just this, sitting on all fours with the nose forward, whereas a human, it's standing upright with the face forward. Okay, and so that incorporates the, like this person here. Okay, that's the anatomical position. So the reference term we use in a human are assuming the human's in that position. But if we instead assume the human's in this position, then all the confusion goes away, right, about the terms. In fact, there are some anatomists who advocate redefining terminology of humans along the lines of quadrupeds, though I don't think that'll ever gain traction. Okay. All right, so those again are the sections, and here we can see some of those sections as applied to a human. So here's a sagittal section in a human, in a MRI, which is a method for imaging the brain, and here we can see a horizontal section. So here you can see the two eyeballs there, can be seen in the horizontal section, and there's the cortex, and then you can see the brain stem coming out the bottom there. You wouldn't see that in the horizontal section in the rat. You would see the kind of brain stem and the spinal cord trailing off down the back, again because the axis is turned 90 degrees. Okay. All right, so next we want to consider the difference between, so again, you need to take a look at these terms and make sure that you understand what they all mean. Next, we want to consider the basic parts of the nervous system, and we have two basic parts. The central nervous system, which is this brain and spinal cord, what we're going to mainly focus on. The brain and spinal cord both develop from the neural tube, as we will see when we look at the pattern of neural development, and can really be considered one structure, one big structure. Now, outside of that, we have the peripheral nervous system, which mainly consists of bundles of axons of neurons that either have their cell bodies in the central nervous system or very near to the central nervous system. So we do have cells that live entirely outside, that have their cell bodies outside the central nervous system, neurons. And in fact, we have some in the autonomic division, autonomic motor division of the peripheral nervous system that have the entire neuron outside the central nervous system, the cell body and all of its processes. Okay? But that's the main distinction we draw. The brain and spinal cord are the central nervous system, and everything outside of that is the peripheral. And we generally divide the peripheral up into sensory and motor. So we have the sensory inputs coming along afferent or sensory nerves, and then the motor output coming along efferent or efferent nerves. And in the case of the motor output, we can divide it between the somatic motor division, which controls the voluntary skeletal muscles, and the autonomic division, which controls the involuntary muscles like the cardiac muscle, smooth muscle, as well as glandular secretions, such as from the adrenal gland. Okay, and we'll get more into that going forward. Now, a convenient way to see that the central nervous system is all one structure is to realize that the meninges completely surround the entire central nervous system. The meninges are a set of coverings that protect and suspend the central nervous system. As I'll show you in a moment, central nervous system tissue is extremely delicate, having a consistency somewhat like jello. And so it needs to be well protected, not only from chemically, as we discussed in the blood-brain barrier earlier, but also physically protected. It's much less robust than a lot of the bone and muscle tissue you would find that's more exposed. Okay, so we know it's enclosed in a bony cavity inside the skull and inside the vertebral column, but it's also enclosed in this set of meninges. And they are the dura mater. Okay, so dura means tough, mater means protector. It literally means mother. But when we say tough mother in 2020, it sounds like we're abbreviating an obscenity. But really, in this context, mater may be better translated as protector. Okay, so the tough protector. Now this consists of a couple layers of dense connective tissue that's extremely tough, extremely hard to tear, but it nevertheless is flexible. It's adhered to the inside of the skull in the brain and attaches to the vertebrae in the vertebral column. There are places where the two layers in the dura mater pull apart. You can see one of these here. They're showing you the dura mater here kind of peeled away, but along the midline there's a space inside the dura mater called the dural sinus, and I will talk about the function of that in a moment. Okay, now deep to the dura mater you have the arachnoid mater, which consists of an arachnoid membrane and then a subarachnoid space. Now in the subarachnoid space you have this filamentous arachnoid material, but mainly what we are concerned with here is that this space in life is inflated or filled with the extracellular fluid of the brain and spinal cord, the cerebrospinal fluid. So the entire brain and spinal cord is surrounded by this liquid. Okay, now if you are completely surrounded by liquid, you are floating, right? So if you go underwater, kind of hang out underwater beneath the surface, you are floating beneath the surface. And so the entire brain and spinal cord is literally floating inside your brain and inside your cranium and vertebral cavity. Now this supports this delicate tissue, sort of like a whale is supported by its buoyancy in water, right? We know whales, when they get on to land, if they beach, they can't breathe well because they can't support their own weight. Okay, so you're similarly, if your brain wasn't floating, it would sort of crush itself under its own weight, have a difficult time for blood to flow through it. Okay, this also provides shock absorption, right? So having that fluid surrounding provides shock absorption. It doesn't work very well for flying into windshields at like 40 miles an hour, but it does work well for everyday accelerations and decelerations that you might experience. Also doesn't work very well for putting on plastic armor and running into 300-pound guys as hard as you can. Doesn't work too well for that either. Which is why when that happens and you overcome the shock absorption ability of that space, right, the brain becomes contused or bruised, and that causes permanent damage. Okay, and then finally, we have the pia mater, which is directly on the surface of the brain. Now, arachnoid means spider mother because of the appearance of it. It kind of looks like cobwebs. And then the pia mater looks like as delicate, very, very fine tissue paper, for example. So pia mater means gentle mother. So it's right on the surface and it follows all the curves. Okay, so here again we can see a picture of the arachnoid mater on the surface. You can't really see the pia mater in that picture, and the dura mater has been cut in a couple places and folded back. All right, I'm going to spin this off, so I'm not making videos that are too long and the thing ends up crashing on me.